Hello everyone. Today we'll be presenting our work in the epidemiological modeling of the Zika virus in Brazil for the 2016 Modsim Challenge on behalf of Team NDSSL. To give some background on the Zika virus, it's a flavivirus transmitted by Aedes mosquitoes and closely related to dengue and yellow fever. It's believed by many to be responsible for the Latin American microcephaly outbreak due to the spatiotemporal correlation in their appearance. It's very strong laboratory evidence and an autopsy study. So, since we started this effort, a couple things have changed, as with any epidemic. There have now been 312 imported cases within the United States, and sexual transmission is appearing significantly more common, with 6 transmitted cases in the US alone. Also, it's now been detected in 33 Latin American countries. So, what we developed was a self-optimizing SCIR metapopulation patch transmission model. And what this is effectively a series of equations that model the transmission of virus between susceptible, exposed, immune, and recovered populations as they travel across a geographic area. During this, we based this on the Manor model, but we selected from their variables only ones that could be expected to change under biological plausibility, ignoring the human death rate, the mosquito infectious transition rate, and biting rates. Also, each experimental variable in this self-optimizing model was seeded with the Manor model parameters for dengue transmission by Aedes aegypti, and our sweep ranges were based on the maximum ranges given for dengue or chikungunya transmission by Aedes aegypti or albopictus. So what this is, it's a simulated annealing algorithm based on the way the protein folds, where a series of small random changes get this linear structure to eventually form a complex conformation that can solve a specific problem. In this case, we wanted to fit these parameters to the Zika epidemic data. The way we did this was we took subsets of the surveillance data from 50, 100, 150, and 200 days, and we trained 25 random models to match each subset. Then, from the best match of those 25 models, we seeded another 25 new models for second stage of optimization, then projected this onto the future curve.
Geospatial analysis has come into play twice in our work. First, we utilized a gravity model to predict the flow of people between patches, and then we used environmental niche models to predict a mosquito exposure in each respective patch. This is the travel network we created. It was stitched together from data from OpenStreetMap, the ADC World Map Digital Atlas, and a hydrogeographic model of the Amazon River Basin. The latter is important because many of the nation's rural regions are inaccessible by road. We then found the population-weighted centroids for each of our patches and calculated the network distance between them. We then fit these data a non-linear Poisson regression trained on known bus travel figures from the Brazilian National Travel Agency. Niche modeling takes known species occurrences and a variety of geographically distributed variables, and then fits regression models to these to predict the likelihood of finding said species in any given area. In our case, we used niche models of Aedes albopictus and Aedes aegypti to predict the number of individuals suffering mosquito bites in any given patch.
Another important piece of our work revolved the use and creation of open data. Data came from the Columbia National Institute of Health, and we used Columbia PDF reports that were cleaned by the BuzzFeed repository, as well as other independent contributors. We cleaned this data to conform to the CDC guidelines and also documented our process in doing so. We also maintain our own version that contains only the CSV data and not the original PDFs. This makes it easier for researchers to download the raw data. Lastly, we created a data visualization for the CDC repository. Data visualization is currently online and to be ready to be viewed and used. This aims to provide a high-level overview of all the data in the CDC repository, including dated entries, number of rows, number of countries, and entries per country. For example, in Colombia, we have the cumulative cases in each city by region, and this gives a means for researchers to quickly view the latest data without downloading the entire CDC repository.
So one of the main deliverables of this project was finding parameters that fit the current outbreak and finding more clinical ways to describe this transmission. So One of the things that was most significant was that we saw a significantly longer human to mosquito infectious stage in this model than from the similar validated models we use as seeds. We also saw a greater mosquito death rate, though we would need to look into this for biological plausibility. For a single population projection on Brazil, we were using data from June 23rd, 2015 to February 12th, 2016, and predicting 12 weeks into the future from the end of the training set. Here, we predicted 274,000 cases of Zika in Brazil by May 6th and 372,000 cases in Brazil by June 3rd. We can also map this on a patched model. So what we're seeing here is the number of susceptible, infectious, and recovered individuals in the exposed populations in Brazil broken down by state and time, but we can show this a cooler way. So what we're looking at here is two different models using the exact same disease but different transmission network. On the left, we have the gravity model network showing spread of the virus from Rio de Janeiro across the rest of Brazil. And on the right, we have the bus network, which is just based on transportation data. So, in conclusion, during this project, we constructed and streamlined a very powerful open source toolchain which has allowed us to predict the course of an outbreak and has allowed the community to directly build upon our efforts. Through this challenge, we provided a very sound rallying point for those with additional talents beyond our own to start to tackle this problem, including adaptive SCIR metapopulation patch models, Zika virus geographic risk maps, and open data epi-surveillance tools. These are the works which we've used in the development of this project, and otherwise, I would like to thank Dr. Brian Lewis, Dr. Stephen Eubank, Dr. Berkey, Dr. Mao, and Dr. Srinivasan Venkatraman for their help in this project.